There's two main types of models, one of which is models that are entirely in mice. They're mice that have an intact immune system, and you can either graft in tumors or things where the mouse is sort of like the human equivalent. It's just that there's no human tissue in them at all. Some people have criticized those because they don't have any human tissue in them, and they have been trying to build what they call humanized mouse models, which typically involve trying to take the human immune system and put that into mice, and then use human cancer cells and see how that immune reaction works in mice. It's very, very difficult to do. A lot of progress has been made in those areas, but one of the tasks in the workshop is to say exactly where those models are at and how they compare to the mouse and mouse. There are a number of models. For instance, it turns out that dogs have a very high rate of getting melanomas. There might even be more dog melanomas than human melanomas in the US. And that's a spontaneous model where the dogs aren't planning or hoping to get, obviously do not want to get melanoma, but you can do clinical trials or look at dogs with melanoma to see if immune therapies could work in it. The problem has been that it takes a whole lot of infrastructure to make that kind of work. That's happened in the mouse research field. In the dog research field for immune therapies, it hasn't happened yet, but it could. And that would be another example of a non-mouse vertebrate animal model for immune therapy development.